An overview, we start with the extractable study. I don't have a slide on the extractable study. All colleagues have presented it sufficiently in depth. But that's where my work starts. Huh? So the study director is very happy he finished this extractables report. He sends it over to the client on time, but the client doesn't know yet if he's happy or not. He wants to know, is it safe? And that's not included in the conclusion of the extractables report. It's also not possible. So I want to share how I tackle such cases when I get an extractables report, how I approach it to make a final assessment. And first of all, you start with a patient exposure assessment. You have done an extract, you have extraction values or concentrations, how much worst case ends up in a patient. And therefore I need to know the production process, all the parameters like batch volume, daily dose, those presentations, and how it was extracted. And that plays also a very important role. Then once we have converted that to micrograms a day, patient exposure, we can compare it with the generic applicable safety thresholds. When they are above that threshold, then we have to qualify it for safety, or alternatively, you're going to demonstrate absence in the drug product itself afterwards. But typically it's a little bit cheaper to demonstrate it with a permissible daily exposure to justify that there's no or little of risk. To come to a permissible daily exposure, there are 3 hurdles to be taken. First one is mutagenicity and carcinogenicity. Second hurdle is irritation and sensitization. And last hurdle is systemic toxicity, which the data you can use for repeat dose studies to come up with a final PDE. And I will conclude with some example cases and take-home messages. So first, extrapolate this extractables data into worst-case patient exposure. There's worst case and there's worst case. So what is reasonably be expected to be migrating and ending up in the drug product itself? So typically you get results in microgram per square centimeters, microgram per unit, how many units extracted, and how does it end up in your final drug product. We have to assume that all extractables which are observed are immediately released. I heard already some discussions about that. But this is FDA wording. In absence of release kinetics, you have to assume everything ends up immediately in the next step of the production. Units are surfaces extracted versus the actual production process. You have to recalculate if the tubing was cut and how long the tubing is, was the right diameter of the process versus what was extracted. So you have to recalculate the actual production component, all these extractables from the contact site, which can be released into the patient. Then there's this dilution factor. If you have a tubing to a mixing tank where the whole batch is coming together, so you can assume reasonably that these extractables are homogenized in that whole batch. What is important, and I put this danger sign there, if you are at the end, downstream before fill and finish, and you have a surge tank, and you're not going to wait until the whole batch is there before you start fill and finish. So there's a minimum volume before you start to fill and finish. So the tubing, or also the sterilizing filter extractables, they are concentrated in the minimum volume before you start with fill and finish. So that's maybe something the production engineer is not aware of. But if you say we can start filling once we reach 100 ml in the surge tank or 1 liter, your exposure will be 10 times higher if you start filling with 100 ml. How do you further calculate it? It's pretty straightforward. You obtain your extractables concentration in microgram per ml before fill and finish, and you multiply it with the maximum daily dose. And then you have your exposure, worst case, in micrograms a day. Are all extraction solvents relevant? That's also an assessment you can make. Typically we do that for medical devices, but that knowledge you can also apply for extraction of single-use systems. And the question was raised, I believe, yesterday, pH 10 versus 13. In the end, you want to have a predictable extractables data set which is reasonable to become a leachable. So, for instance, if you see at a very high pH of a filter polycarbonate housing, you see bisphenol A coming out, which you should be considering then. But if you have then, let's say a BPOC, an older report, where also the neutral pH was included, then you can evaluate in that extract, is bisphenol still present there, yes or no? So if it's not present, you can reasonably assume that if you bracket the pH between 3 and 5.5, let's say, for a drug product of 4.8, it's not reasonable to expect that bisphenol A will migrate into the drug product. So that's also a consideration if you want to qualify all your components for multiple drug products at multiple pHs. Maybe taking the neutral pH as an additional solvent can help you waive the high pH or the low pH, depending on the pH of your product. Then we compare the extractable levels in micrograms a day with the applicable safety thresholds, which Karen also already touched upon. So this is the ICH M7, and for chronic therapies, more than 10 years, you see it's 1.5 micrograms a day. And for shorter therapies, like for instance, an antibiotic, you can say up until 120 micrograms a day is still acceptable. But that's only valid for mutagenic impurities. I will come back to that later. How does this stage approach was established? You see the graph there. It's based on Haber rule, which FDA doesn't accept or doesn't like to be applied if you go from a lifetime PDE to a short duration therapy PDE. So the Haber rule says if you have a concentration C1 and a duration of exposure T1, the toxicological output is the same if you have the half of the concentration for the double amount of time. It's a constant. So the dose is also the poison. Imagine, we take the example of ICH M7, 1.5 micrograms a day. It's good for lifetime exposure, 70 years, 25,550 days of exposure is equal then 1 day having 38.3 milligrams. So all vaccine producers would be very happy to see that. This is unfortunately not the case. So they apply additional safety factors. You can see at the short, less than 1 month, they go from 38 milligrams to 120 micrograms, applying a safety factor of 300. That's also something you can consider applying to lifetime PDEs, coming back to a short-term permissible daily exposure rather than lifetime. This 120, I don't see it often anymore, but a few years back, clients instructed some study directors, oh, we have a short therapy. I want the AET calculated based on 120 micrograms a day. That will not fly with FDA. This is the PQRI. They have the same concept, safety concern threshold. And there you see also the lowest safety concern threshold is also 1.5 micrograms a day. They don't put any duration to their classification. Sensitizers and irritants, they have to be controlled at 5 micrograms a day, which is also the qualification threshold stipulated in the most recent PQRI. So everything above 5 needs to be identified and evaluated for safety. For mutagenicity, for short term, you can go higher, but 5 micrograms a day, then if that happens, you have to go to the toxicologist. There is no generic alternative threshold like there used to be. If it's not mutagenic, not sensitizing, not irritating, you had 150 in earlier days as a generic safety threshold. Then it became 50. 50 was proposed, is not withheld. LC comes up with 35. I'm not using that yet without being accepted. So the only next threshold you have after 5 micrograms a day is the permissible daily exposure. What I also want to stress is that the 1.5 micrograms a day, that you use that value in determining your analytical AET. That's the preferred. Let the toxicologist afterwards evaluate the data itself, but screen as low as possible. So the conclusion is, for chronic therapies, 1.5 micrograms a day is a threshold. For non-chronic therapies, you can go to 5 micrograms a day. So, initiating the safety assessment, you have done your exposure assessment, maximum daily exposure, and also knowing the length of therapy using 1.5 or 5 micrograms a day. So you look at the report. Sometimes it's a Nelson Labs or a competitor or a supplier report. The first thing you have to do is evaluate, for sure, if you have a supplier report, is this reporting limit low enough to cover your application? Sometimes I've seen, reporting limit is 1 ppm. Okay, all right. But you don't know how much square centimeters was extracted in 10 ml or in 100 ml. So you cannot recalculate that thing. So this is important that you screened or the data screened low enough. Sometimes I've seen even no reporting limit, and then you taken under assumption the lowest concentration reported, you take then as the reporting limit. Select the highest levels across different techniques. So the techniques can have overlaps in detecting certain extractables in a different solvents and different time points. So we always rank all the highest worst case exposures. And then you assess all these compounds which are above 1.5 for chronic or above 5 for non-chronic therapies. Like mentioned, there are 3 hurdles to overcome. Mutagenicity for chronic start with 1.5, sensitization and irritation from 5, and general toxicity also from 5, but that would lead then to a permissible daily exposure. The term, mutagenicity, and the broader term is genotoxicity. That is at the micro DNA level, and clastogenicity is at the higher macro level, where you have like chromosomal aberration, where chromosomes break off, or where the cells doesn't split or duplicate all of the DNA material equally. These are threshold mechanisms, meaning that you have to be exposed for long term at higher levels before it becomes carcinogenic. Genotoxicity, specifically at the DNA level, is referred to as mutagenicity. It does not have a threshold level. That's why the level is so low, 1.5 micrograms a day. And that is also the focus of ICH-M7. They say mutagenic impurities. And this is what happens at the DNA level. The golden standard is the Ames assay, bacterial reverse mutation assay. So that data is what you want to look for. Since it's so an important assay, I will briefly discuss the principle. So what you do is you have a Petri dish, and you have at least 5 different strains of Salmonella and 1 E. coli, which have a gene knocked out, so they become histidine dependent. They cannot grow without histidine. The repair mechanism, when DNA is copied and base pairs are not copied correctly, normally you have a repair mechanism that corrects these false entries. So it's a very susceptible microorganism to detect at a high sensitivity level these mutations. So you put the extractable together with these microorganisms and a little bit of histidine, because remember, they need to be able to grow. They need histidine. And you can do it without and with S9 mix, which is liver homogenate containing hepatocytes, microsomes, containing the CYP enzymes for the metabolism of this extractable. So maybe the extractable itself is not mutagenic, but when it becomes metabolized, it could become mutagenic. So then you incubate these plates, and if they don't grow, then it's fine. If they are able to grow, means that after some cell divisions, they mutated back because they no longer need histidine to continue to grow. So if you see colonies at your plate, they were back or reverse mutated and no longer need histidine, and then you consider them as mutagenic. So when it's positive for mutagenicity, then the likelihood is higher that you can also find carcinogenicity data. This is the TD50 you would be looking for. So that's the output of the endpoint of a carcinogenicity study. It's a 2-year study typically in rats, where you define the dose upon which 50% of the test animals develop tumors. That concentration or that exposure in milligram per kilogram, that's the data you want to have. And sometimes you have multiple carcinogenicity studies. So from a conservative point of view, you take the lowest concentration for which this TD50 is observed. And then you do linear extrapolation. As an example, it's coming from ICH Q3C, by the way, for 1-chloronitrobenzene, the TD50 is 117 milligrams per kilogram a day in female rats. So how do you extrapolate that to a human safe dose? So they divide the TD50 by 50,000. Why? Remember, the acceptable risk to have or to develop cancer is 1 in 100,000. TD50, 1 in 2 develops a tumor. So to bring that to a chance of 1 in 100,000, you divide by 50,000. And then it's expressed in milligram per kilogram times 50 kilograms a day. You end up with 117 micrograms a day, which is an acceptable intake for mutagenic or carcinogenic impurity. If it's negative, then you can proceed to hurdle 2. Mutagenicity is not of an issue, but most of the time we don't intentionally put leachables into a filter or a tubing or whatever. So these are unstudied chemicals. So often there is no toxicological information. These impurities are also not produced in 10,000 tons, where the REACH regulation requires you to do a full toxicity evaluations. So what you do then is you are allowed by ICH M7 to revert to QSAR methodology. So that's structure-activity relationships. These are software models, artificial intelligence of all a letter, I would say. It's 30 years ago they started with it, with databases which have information about 9 of 10,000 compounds with mutagenicity information. An example here is the output for the rubber oligomer, the non-halogenated and the chlorinated. So the non-halogenated is negative in Derrick and multicase. Derrick is a rule-based system. Multicase is a statistical-based system. You need to have 2 complementary systems with the same prediction to be reliable. Why did we do Derrick and multicase? We've done an exercise internally back in 2015, I believe, where we compared also with, and we took about 20 compounds and we checked which compounds are most in line. And then we went for these 2 models. I was in a congress in 2016 on QSARs, and the question was raised, who does all do QSARs? And one big pharma company said, we're running all QSARs, all available QSARs. So the more prediction systems, models you run, the more likely you will have conflicting conclusions. And then what you do? You take only the negative predictions for mutagenicity and put that into your package. So that's not the kind of service we wanted to offer. So we stick with these 2, but of course there are other alternatives you can evaluate. The halogenated turned out to be plausible for mutagenicity, so there is more evidence that it would be mutagenic than it wouldn't be mutagenic, and for multicase it was positive. So maybe also very briefly the difference between a rule-based and a statistical-based. Rule-based is knowledge-based. You see a substructural feature, a functional group, which is known to be causing mutagenicity, so it's classified as mutagenic. Statistical-based, you look at the whole molecule and you have a data set of molecules all containing the substructural features, and the statistics will tell them out of 100 compounds how many with that structure, that functional group, will lead to a positive mutagenicity assay. So that's based on statistical processing. And you have to have both. So if you have run both of them, you come into the classification of ICH M7, which also allows to say in which condition you continue with your assessment. So class 1 is mutagenic carcinogen, so you know it's mutagenic, you know it's a carcinogen. So there is obviously TD50 information, so you can use the TD50 value to derive your acceptable intake. If it's a known mutagen and you don't know if it's a carcinogen, yes or no, what you can consider is to do a read across with another carcinogenic compound. Read across is not always accepted. It has to be well justified, but it's basing your PDE or acceptable intake based on a highly similar compound or multiple similar compounds if one compound doesn't contain all these substructural features. Class 3 is alerting structure unrelated to the drug substance, so that's can be that's applicable to extractables and leachables. So it's positive. Or you control it at the TTC, the ICH M7, or you can try to overrule it. How you can overrule it? Actually by performing the Ames assay. If it's Ames negative, you go to class 5. If it's positive, you have to do probably then the read across. If both systems predict negative, then you can go to a higher threshold, the permissible daily exposure based on an OEL, or maybe you can still use 5 as a qualification threshold if it's between 1.5 and 5. It's a narrow window, I know. Or if there is no data, it's not mutagenic, there's no data, you can still revert to the read across approach. Hurdle 2, that's the 5 micrograms a day, irritation and sensitization. Irritation is reversible upon skin contact. You get a rash development, but it goes away after time. Corrosion, then there's really necrosis of the dermis, and it will lead to scar tissue. The OECD 404 is the test you want to look for to see if there's data, but the overall concentrations below 1% in the CLP regulation are considered, or mixture are considered not to be irritating. So extractable leachables rarely come at the level of 1%. So typically irritation is not considered as a concern. Sensitization, that's a substance that will induce an allergic response upon following repeat contact. It's a very complex endpoint to assess. It's a cascade of biological processes, and it's very hard to put thresholds to these levels. So 5 micrograms a day is the level for sensitization. What happens is your leachable will enter your body, but an antibody will never detect a small molecule. Small molecule is as big as this thing, and a protein will be as big as this room. So antibodies don't detect it. So what needs to happen is that this leachable reacts with the protein. Then it can an antibody can detect it as this is an intruder. Your system develops an antibody. This is the induction phase of sensitization, and then you have the elicitation phase where you have repeat exposure. These T cells have a memory function. They know this intruder. Next time you're exposed, they release an army of antibodies and a huge immune response, which goes together with fever, even anaphylactic shock, which is considered as a very critical endpoint. The test you want to look for is the local lymph node assay, which is validated to assess the sensitization potential. An alternative test, also mainly used for medical devices, the guinea pig maximization test. But if I have to run a study, if I cannot waive sensitization, I typically run a local lymph node study. So it's an in vivo study. So you have a mice, and you have very thin ears, and you put left and right 25 microliters, the highest concentration possible, and you do it for 3 consecutive days to have this repeat exposure. After day 6, you inject a radioactive label targeting the lymph nodes, and after injection of this radio label, 5 hours later, you retrieve the lymph nodes, and you're gonna analyze it with a liquid scintillation counter to measure the amount of radioactivity. If the radioactivity is 3 times more than your control, which didn't include the leachable, then it's considered to be sensitizing. And the EC3 value is the percentage, the dose level, the concentration at which the stimuli index is 3, thus from which level something becomes sensitizing. LC framework had a recent publication also on the evaluation of sensitization. They suggest to focus on the strong and extreme sensitizer. It used to be in the PQRI in earlier days. It's for hyposensitizing compounds, this 5 micrograms a day. It dropped off. I don't think they forgot to delete it. It's probably on purpose. So they put just 5 micrograms a day as a limit for sensitizer. But also the PQRI opened the door for QSARs. In earlier days, it was not allowed to use QSAR prediction models to predict sensitization, only mutagenicity. But nevertheless, we always include it as additional weight of evidence, if you wish. But also the LC framework proposes to include the QSARs. Also evaluate the reactivity of a molecule, like mentioned, to have this hapten formation. Can ask a medicinal chemist how reactive is this leachable, but there are also QSAR tools again, like OECD Toolbox, where you can predict the reactivity of a molecule. And if the outcome is moderate or weak sensitizing properties, you can consider it as a negligible risk. And you can proceed to hurdle 3. Just to give you an idea about these categories of the EC3 values. So these are concentrations. The extreme concentration of extreme sensitizer has to reach a stimuli index of 3 below 100 milligram per 100 ml, so 1 milligram per liter, I believe. So it's already a relative high level before you can consider it as an extreme sensitizer. If QSARs predict that there is a sensitizing potential, Derek has EC3 values predicted, multi-case doesn't. But if it's a sensitizer, you can consider to overrule it again, like with the Ames assay, you can overrule the QSARs. With the local lymph node, you can overrule the QSAR. And I use this classification of potency to select the doses of the mice in the local lymph node assay. You have to start with the highest dose, where no irritation, it has to be soluble, homogenic, where no irritation and systemic toxicity is observed. The ears cannot swell, they cannot lose weight. So that's the highest level. And then you take 1% and 0.1% to be sure you can discriminate between a moderate and a strong sensitizer. Then you can apply the justification, if you wish, of the LC framework. Imagine mutagenicity and sensitization is overruled. Then you have to develop a PDE value, and then you're going to look for, the NOEL value. What is NOEL? No observed effect, adverse effect level. So if you do a repeat dose toxicity study, you have 3 levels. The highest level has to induce toxicity. The lowest level should be no adverse effects. So you want to observe this NOEL concentration. It's not a wild guess. A lot of dose range finding studies go before you go into a full repeat dose study to come up with this NOEL. But multiple studies have been performed typically, and also you take conservatively the lowest NOEL observed in a repeat dose for that compound. And then you can calculate the PDE, which is nicely described in the ICH Q3C. So you take the NOEL in milligram per kilogram. You multiply it by the weight of the person, and then you take additional safety factors. If level safe for a rat is not a level safe for a human. So F1 is compensating for the extrapolation from different species. If you go from a mice to a human, I think it's dividing by 12. If you go from a dog to a human, I think it's 5. So to compensate that. Factor 2 is variation between individuals. I may respond differently on a chemical than one of you guys. So that's a standard value of 10. Then the duration of the toxicity study you took as a point of department compared to the chronic exposure during the therapy. That has to be compensated. If there is any unusual toxic effect like teratogenicity, so that bijvoorbeeld the reproductive systems are affected, that the pups are affected, if you're breastfeeding the pups, that it is affected, neurotoxicity, non-genotoxic carcinogens, remember the threshold ones, that's an additional factor you can put in there. Also, the point of departure, sometimes you don't have a no adverse effect level, but only the lowest observed effect level. Then you have to divide at least again by 10. So then you come up with a very conservative PDE. Then ICH Q3D also says, well, this is for oral PDE calculations. In this case, we are talking about injectables. So if you have an oral dose, how much of this compound will go into systemic circulation, into the bloodstream, affecting all organs? So that's something you have to take into account. How much is bioavailable? So that's a peak a pharmacokinetic study. So you give oral this leachable, and you take blood and you see how much goes into the bloodstream. So that's the bioavailability. As an example, take bioavailability above 50%. So more than 50% of your oral intake goes systemic. So imagine you have a PDE oral 100 milligrams a day, but that was an oral study, and you know only 50%, 50 milligrams go systemic, causing all this organ toxicity. So this is also the level applicable for an IV PDE. There's also being discussed now factor F7 for read across and other uncertainties you wanna have included. So it becomes more and more conservative, unfortunately. Typical feedback you might receive on your extractables or safety evaluation being sent to FDA or any other authority. The leachables do not have CAS number, indicate the validity of the assigned structures. So if you're not having a confirmed structure, most probable or related to or whatever, and you do then a QSAR, then it's an uncertainty about the identification, and it's a prediction, also an uncertainty by QSAR, that rarely will be accepted. So identification is a must. Other than mutagenicity, QSAR assessments for respiratory, skin sensitization, etc. Is not sufficiently validated. That is now hopefully off the table, and we can systematically have QSARs accepted as a prediction for sensitization. Sometimes they don't want to have this feedback. If they don't follow your reasoning in a read-across approach, they say, well, find data. If the data is not available, run a toxicity study. A 14-day tox study in one species using isolated leachables. That's a challenge. Or the API enriched with the identified leachable. Then you have to have it synthesized as well. administered in a clinically relevant manner at leachable levels equivalent or greater than what the patient would be administered. I've seen this already a few times. Only had to run an animal study on one compound. That will become on the next slide. For chemicals identified as an extractable only, develop analytical methods with higher sensitivity such that the absence of the extractable in the proposed drug products can be confirmed at lower levels to assure that the daily exposure will not exceed 5 micrograms a day. This is a relevant observation and for the 665. So when we do this worst case, everything is released immediately from the component. It ends up in the patient. It's above 5 micrograms a day. We have this PDE, let's say, of 800 micrograms a day. And worst case, we calculate it's 10 micrograms a day. What is the added value in developing an analytical method showing absence of this compound at 5 micrograms a day in the final drug product? There is no added patient safety value. It's only a loss of time and money and effort on our side. Then, since your root cause analysis identified a mutagenic compound X as an ingredient of an ink in this example, of a primary film of a bag, you can address this efficiency by removing the ink from the proposed products and replacing it with another one. Regularly, I get, without context, the request, prepare me a permissible daily exposure. It's mutagenic, we know. So you prepare the best way you can a conservative permissible daily exposure, and it's not accepted. Why? You need to know where your mutagenic impurity is coming from. ICH-M7 is control of mutagenic impurities. If you do not know the source of your mutagenic impurity, you can hardly be in control of that impurity. So that's very important. And even if it's there, they can also ask you to remove it to reduce these mutagenic impurities from your product. Last example I have here, for the identified leachables containing API fragments, you applied, that was me, ICH Q3B qualification threshold of 0.15%. So in this case, the rubber oligomer formed an adduct with an API degradation product. These adducts are not neat degradation products of the API, but are forming due to the interaction of the API or API degradation product with the rubber closure. The ICH Q3B limits do not apply for the safety of the leachables containing the API reaction adducts. This was my first attempt to qualify that adduct, because in the ICH Q3B, and it's still there, it is listed for degradation products, but also excipients and the reaction with primary packaging. It is in there. So I thought, we're good to go. We're using this 0.15%. It was not accepted. Then we did QSARs on the whole adduct, on the separate, the two portions of the molecule, the rubber oligomer, the API degradation product. Qualified it or presented as a PDE, was not accepted. That's the one time I had to do a rat study. The rat study was not convincing enough because if you do an animal study, it's like going into clinical trials because if you're looking for tox data, it's all reported in milligram per kilogram, all these NOALs. If you're going into clinical trials, you have to not use the same milligram per kilogram, you have to use the human equivalent dose. And that takes the evaluation also of that takes the body surface into account rather than the weight. Then take-home messages. The dose is the poison. That's a given. The NOAL levels in systemic toxicity studies you can take for PDE only if it's not mutagenic, carcinogenic, sensitizing, or irritating. Conservative approach taken for mutagenic impurities if it's mutagenic, it's not a disaster yet. You can find perhaps this TD50 values and do a linear extrapolation, which showed that you came up with a higher tolerable intake. Or you can take the stage TTC going up to 120 micrograms a day, but that's only for mutagenicity. Yeah? So that stage TTC you cannot use as an identification threshold. Keep that at your 1.5 micrograms a day. And if there is data on a leachable or an extractable for that matter, use the data rather than going to QSARs. Data overrules QSARs. Also, generic thresholds are very low, so maybe with the data you can set a higher safety threshold, which shows a higher margin of safety when you submit your data. Last point, leave toxicology to the toxicologist. All assessments should be verified by a certified toxicologist. I think it's a good approach. Me, myself, I'm not a certified toxicologist, but I consider myself as an expert after 10 years. But it's good that you have a second opinion, reviewing. Did you miss something? Are you sufficiently conservative? Because if you ask 10 toxicologists about a PDE, you will get probably 5 different values. Hereby I conclude my presentation. Thank you for your attention.